types of useful polymorphic DNA sequences. First one, RFLP, restriction fragment length polymorphism. Second one, variable number tandem repeats 10 to 50 base pairs, that is VNTR, short tandem repeats 1 to 10 base pairs, that is STR, single nucleotide polymorphisms, SNPs. The process of determining an individual's genetic polymorphisms is known as genotyping. Many tools for assessing DNA polymorphisms are now available. Some of these methods assess the length polymorphism, intels, sequence polymorphism, base changes or rearrangements, or combinations of the two. DNA sequencing reveals all types of polymorphism, but is costly and labor intensive. Gel electrophoresis is used in DNA analysis to both separate and sequence DNA. PCR is frequently employed beforehand to produce large quantities of the DNA to be analyzed. SNPs are very useful for defining Y chromosomes or mitochondrial DNA haplotypes and DNA phenotyping. Restriction fragment length polymorphisms can be separated and identified by gel electrophoresis. A single nucleotide polymorphisms A to C causes loss of a restriction site. The importance of SNPs in DNA typing. DNA typing is also called DNA fingerprinting or DNA profiling, is a procedure wherein DNA extracted from a biological sample obtained from an individual is analyzed. The DNA is processed to generate a pattern for each person is termed as a DNA profile. This profile is unique for each person excepting that derived from identical twins. The scientific basis of DNA typing. DNA typing is founded on a number of genetic and molecular principles. The concept of the cell is the basic understanding of the complexity of DNA typing. A cell is the building unit of an organism made up of its component parts, one of which is the nucleus that functions as its command center. The nucleus houses the DNA that codes for the genetic information responsible for all cellular processes. Several DNA molecules comprise genes which in turn are located in minute bodies called chromosomes. In humans, there are 23 pairs of chromosomes within a cell, thus making up a total of 46 chromosomes. Some chromosomal regions contain repeating units, that is short tandem repeats or STR markers of the same type of DNA molecule, which may or may not code for a specific protein, have been used as markers for human identification in forensic casework. DNA can also be found in another part of the cell called the mitochondrion. Unlike nuclear DNA with 2 copies per cell, multiple copies of mitochondrial DNA up to 1 lakh copies are present per human cell. Because of this, mitochondrial DNA analysis is the method of choice when dealing with environmentally challenged samples. Example, identification of mass disaster victims, exhumed human remains, etc.